So this is about a project that I currently work on at DZ, indeed for which I was hired to work on, which is called Amarcord, and it's supposed to enable extended collaborative research in a field called crystallography. Now, I can't assume that you know what crystallography is, so I'm going to just quickly explain how we use it here at DZ. So the idea of crystallography, or specifically X-ray crystallography, what we do here is to create images on an atomic scale using X-rays. Atomic scale meaning proteins, bacteria, molecules, that sort of stuff. We use X-rays. That means we need to have an X-ray generator. And one of our X-ray generators is here. It's called PETA-3, which is actually a particle accelerator. But the acceleration of particles isn't really interesting for crystallography. It's really the deceleration, because when you decelerate these particles, they give off their excess energy in the form of X-rays, pretty strong X-rays, actually. So the idea here is that when you decelerate, for example, the particles, which in our case are electrons, for example, here in the experimental hall, you get sort of tangentially to this circle, you get X-ray radiation, which then goes through, on the top right here, through this narrow tube, and then emanates on the other end, and you can use it to shoot stuff with. And it's called crystallography. So what we're shooting is actually crystals. Now you might think that, well, if you want to take images of proteins, why don't you shoot the proteins themselves? Well, you can do that, but the idea is that, unfortunately, we're not there yet. So you can produce images with that, but these images aren't good enough to really be used to analyze the proteins. So instead, you take the protein and you grow a crystal from it, sort of like when you have salt water and you evaporate it, you get salt crystals. You might be able to get protein crystals. And as you can see here on the bottom photo here, that's the beam line where these beams emanate in the end. The beam comes from here, then hits a crystal, which is too small to be shown here. This one is cryo-cooled from the top right, and then there is a detector at the other end, which you can see here. And the images you get look sort of like this, which might look strange, but then what do you expect from these scale, so to speak? You have to trust me that there is software that is able to interpret this weird image. Specifically, you might see these black dots, and the arrangement and the size and the intensity of these dots can then lead to an image of a protein, for example. Okay, so why am I talking about this? Because you can actually use this to discover new drugs. Why would you want to discover drugs? Well, maybe if there's a new virus going around and you want to find drugs that work against it. And that's exactly what the DZ scientists did when they sort of were hit by Corona in the beginning of 2020. What they set out to do was a sort of screening pipeline. And the idea of a screening pipeline is relatively simple. You take your target protein, which is, in our case, a part of a coronavirus here, so SARS-CoV-2, and you not only put that into a crystal, but also, in addition to that, some compound. And this could be a drug that's already on the market, or a drug that is in late-stage trials, so will be on the market soon, or simply a compound that you know, like caffeine, for example. So you have your crystals that are produced by biologists in the lab, and that takes on the order of days to months, depending on if you're lucky, basically, or how you can refine your protocol for growing the crystals. These crystals with, both these things in it will then be transported to the beamlines that we just talked about, and there will be screened. And at the beamlines, there are beamline scientists who know their tools, but maybe know not a lot about biology, so different kind of user group. And one of the most important information about crystallography in the slide is that this screening actually takes minutes. So if you can sort of parallelize everything else, screening can be done very efficiently. The screen crystals will result in images, just like the one we saw just now. And another group of people, namely structural biologists, who again know their biology and chemistry, but can also work on a data cluster that we have here, which is called Maxwell at DZ. And these take then, well, hours, and depending on how well you know the stuff you're shooting at, can also take months. And they will generate an atomic model. And I said this is for drug discovery. And the idea is that in this final atomic model, you will actually, of course, see the part of the virus and your compound, ideally. And you will see them in sort of a bound state. So that the compound sort of attached to the virus. which could mean that it actually inhibits the virus. Doesn't have to be, but it's the first step towards that. Now, what you can also see is this software that I'm working on is a database, as you can see by the symbol, and it also is involved with all of these user groups and during the whole pipeline. So the goals were to actually link all these activities from crystallization to constructing the final atomic model, to record metadata and do this automatically if possible, because like humans entering stuff make mistakes, machines sometimes also, but not so much. As you saw, there are biologists and beamline scientists. We cannot assume anything about how much they like Unix command line interfaces. So what we needed to build was a sort of usable user interface for all the groups, so they could easily participate in the screening campaign. And the end goal would be to enable concurrent, remote, transparent work on a project with many participants. Concurrent, as you saw, like in a pipeline, that's a given. Like crystals are grown while other crystals that are finished are being screened, while other screened results are being put into atomic models. Remote work in Corona times is a no-brainer, I guess. And it should also be transparent, because one of the most common mistakes that is being done with these things is that people analyze data more than once because they didn't know that somebody else was working on it, or they cannot speak to the person who grew the crystal because they don't know who this is, and that we aim to fix. And usually, like the usual approach for these scientists was to use Google Spreadsheets or Excel. And sure, you can do this up until you have a certain number of participants or a certain number of rows. And even if you do that, Excel spreadsheets tend to not like the concurrency, and there will be data loss. So in this case, the DAISY people were smart, and they asked the research software engineers, so people like us, so what would you use instead, if not Excel? And the answer is, of course, some sort of relational database, right? And its maiden voyage was already completed, and as you can see, there was a paper, and there were lots of people involved and lots of groups involved. I hope I got them all and with the right acronym. The names here in bold are actually 4 people that really worked on the database, so they were primarily involved with that, whereas the other people sort of used it. So you don't need a lot of people to make such a big collaboration work. And technically speaking, there are only 1 or 2 people really working full-time on the database. So that was quite successful, I would say. With respect to technologies, it's pretty standard. We used Python version 3, PyQt for the user interface, because that's what we knew. Same for MySQL, no specific reason to use that. Could have been Postgres or any other SQL database, maybe even NoSQL. And we replicated this onto nodes just to be sure. So it's more about data loss here. So the user interface, due to time constraints, is mostly table-based, but at least there's some symmetry in this. So on the left, you have, in this case, for the people that grow crystals, you have a list of already entered crystals. As you can see, every crystal has a primary key, which is this generated ID. And on the right, usually you have a form where you enter, who are you? What did you just fish from this protein solution, and what was in it? And then the people at the beamline, once this stuff has been transported there, have a similar user interface in the sense that they also have this crystal ID. I mean, that's the key that sort of from the beginning to the end will be the identifier. But they have different columns. For example, was this a successful diffraction image? So was the image sort of not blurry or whatever? And maybe even a comment as to how good this image was and what the resolution was in angstrom. And on the right, again, a form to add a new diffraction if you just hit it with a beam. And the analysis people, by now you know this, also have a table, but this one is sort of a super table because you can choose which columns you want to display, and you can also have filters and all of that. Yeah, that's basically it. So regarding statistics, how was this used and for what? So it was mostly used, of course, in April and March of 2020, and then mostly on Corona. There was another big swath of crystals coming in December. That was a different part of the virus. But as you can also see, this was continually used from that point on. It's not really current data, but it's still being used. So if you're not a biologist, and I am neither, you don't know what this is, but this is a part of the virus, the main protease. This is another part of the coronavirus. This is also a part of the coronavirus. This isn't. This is actually something different, but since these screening pipelines are so common at DAISY, people just reuse the software because it's not really hard-coded to corona. I mean, it has corona in the database name, but other than that, it's totally fine to be used in general. And I mean, that's impressive results already. I mean, there's 17,000 compounds, which are drugs or compounds in general, 14,000 crystals, which were all like entered by hand. There's no robots doing this yet. And then 15,000 images, 43,000 data processing results, and that's a lot, but that's automatically generated. These data processing things, you can have like scripts that generate them. Same for the refinement results, so like closer to the atomic model. And there's more than 100 people involved in the whole thing, so that's quite a thing.